All right, so we're going to go ahead and get started. People are still joining in, but thanks everybody for being here on time. My name is Nick Phelps. I'm MACERC Director and also a faculty member in the Fisheries, Wildlife, and Conservation Biology Department at the University of Minnesota. Before I introduce the speaker for this session, just a reminder that the questions can be put into the chat box or the Q&A box in the bottom of your screen. You can type them in as you go, but we'll save the questions until the end and Meg can address them then. So it's my pleasure to introduce Meg McCachran. She is a PhD student in my lab working on the risk of baitfish as a pathway of invasive species and pathogens, primarily here in Minnesota. Meg, you can go ahead and get started.
Awesome. Thank you, Nick. Yeah, as Nick said, my name is Meg, and I'm going to be talking to you today about my PhD research, which we have been doing for the last 2.5 years and have plans for the next 2 years. So I'll talk a little bit about what we've done so far and touch on where we're hoping to take it next. The title of my talk is Small Fish, Big Problem, Quantifying the Risk of Pathogen Introduction via the Live Baitfish Pathway. And this is work done not only by myself, but also by Nick, as well as 2 members of the Center for Animal Health and Food Safety here on campus, Kata Picasso and Jan Mladanicki. So special thanks to them. I'm just trying to advance here. Here we go. Okay, there we go. So for those of you who are not familiar, the use of live bait is extremely popular in Minnesota. We have hundreds of thousands of anglers that go out every year, and a large majority of them choose to use live bait. For a majority of those folks, minnows are the most popular. So small fish such as this one seen here that are often harvested from the wild or raised on farms, transported to retail bait shops where they're purchased by anglers, and then taken to the bodies of water where the anglers are fishing, and may inadvertently or on purpose be released into the water when the angler is done. So this industry is not only very large and important economically with a $2.4 million bait industry, but it also supports an even larger recreational fishing industry. But this benefit of live bait use in the state is sort of balanced against the possible risk of invasive species spread along this pathway. This is due in large part to the high volume, minimal testing, and low ability for us to trace these fish as they move across the landscape, along with any pathogens or aquatic invasive species that might be present in them. Recently, the pressure has increased on this particular issue because there are perceived and actual minnow shortages. So there is a high demand for bait that is legal and is able to be used in the state. But there's also a lot of question about how do we procure a safe, reliable bait source for all these anglers that want to use live bait, but don't want to spread invasive species or pathogens that could be also present in the baitfish species. So as you can imagine, lots of complexity, lots of high interest, lots of passion leads to a really tangled mess as far as understanding the risks and benefits that this pathway provides. So the question that my project is focusing on is what's the actual risk and what should we do about it? In order to understand the risk of a pathway, we have to think about all the steps that have to occur for a pathogen to spread along the baitfish pathway from an angler to the lake where they might be releasing their bait. First, the pathogen has to be present in the bait species, whether it was sourced from the wild or raised on farms. An angler has to buy those fish and take them in their bait bucket or their live well to their destination. And then crucially, those fish have to be released into the water. All of these steps in the pathway contribute to risk, and I'm going to tell you exactly how we quantified each one and how they each contribute to the risk. This first stage of the pathway, figuring out which pathogens might be present in the bait species of interest, is going to be the first kind of third of my talk, talking about identifying the hazards. What's out there and what do we need to be concerned about? And secondly, I'm going to talk about how we quantify the behaviors that actually perpetuate that risk along the pathway. How are anglers behaving and how many of them are doing that? And what does that have to do with the risk overall? First, I'm going to talk about identifying the hazards. In general, a hazard is a biological organism that can cause disease in the organism of interest. A recent survey of Minnesota bait shops found a number of viruses, parasites, and bacteria that were present in golden shiners and other bait species. And this is true in a lot of jurisdictions, not just Minnesota. Baitfish have a lot of stuff swimming around with them. But that doesn't mean that all parasites, all viruses, all bacteria present are going to be problematic. So how do we sort through which pathogens are the ones we should be concerned about and which ones are just simply hangers-on that don't actually have any effect on the fish? On one end of the spectrum, we can do nothing, and we can do only a reactive management of outbreaks after the fact. We can allow all species of bait in all ways with no bans on particular sources or species. On the other end of the spectrum, we could test for every single pathogen known, and we could do the far extreme and ban all live bait. I think for fairly obvious reasons, both of these extremes are not really preferable. They're extremely burdensome on regulators and producers, but also at the other end of the spectrum, risk allowing a great amount of harm to our wild fish species. So we seek to strike a balance in the middle and do evidence-based hazard prioritization to balance the benefits of live bait use with the risks that could come with it. And to do this, we want to identify and target the most hazardous pathogens, or the pathogens that are most likely to be transmitted in live bait and cause harm to fish species of interest. So that's what we did with the first part of my project. We created this screening tool that allows one to filter from all fish pathogens, every virus, bacteria, parasite out there, using this series of questions, figure out if it's something we should be concerned about. So as you can see, if a pathogen is not considered hazardous to fish by any of the national or local agencies, if it can be carried by baitfish species, that's our pathway of interest, so that's what we want to care about here. And if fish of concern, fish that we care about, are susceptible, then a pathogen would make its way through this filter and it would be considered for ranking in our hazard ranking exercise. If at any point a pathogen gets knocked off this list, we don't go back for it. It's not something we're considering moving forward. So this allows us to eliminate any pathogens that we don't need to be worried about while maintaining focus on those that we do. But all pathogens aren't created equal. So the second step that we did after initially filtering these pathogens was create a risk scoring system. The way this works is we have 7 criteria: the likelihood of transfer, prevalence in Minnesota baitfish, potential for colonization in Minnesota, the host species that it infects, the economic impacts, the ecological impacts, and its current distribution in Minnesota. And each of these factor into its total risk score. We gave each of these factors a different weight, which allowed for things that experts found really important for determining overall risk to have a greater bearing on the total risk score. So for example, if experts thought that colonization potential had the greatest amount of weight on the score, the particular pathogen's colonization potential would have a greater impact on its risk score. On the other hand, if ecological impact was determined to be the most important criterion, then ecological impact score would have a greater impact on the total risk score. So taken together, we get a total weighted risk score, which is a sum of all the individual criteria scores for each pathogen. And briefly, this is what we found. We have results of this forthcoming in a paper that's under review right now, so stay tuned. But in general, we found that our matrix, our ranking scheme, was able to differentiate between pathogens. The top 3 pathogens that were identified were Asian fish tapeworm, which is a macroparasite slowly making its way across the Great Lakes, Ovipliostophora ovariae, which is a microsporidian parasite of golden shiners, and the famous viral hemorrhagic septicemia virus. So these 3 pathogens identified as the highest risk in our risk ranking scheme have also been identified in other risk ranking schemes as possible pathogens of concern for the live baitfish pathway. So we've done the first step, we've identified the hazards. Now we need to do the second part of actually quantifying the risky behaviors that allow these pathogens to spread from the bait shop to the shore where they may be dumped into the water. It's not enough for the pathogens simply to be present in the bait supply. They have to make their way along the pathway as well. I'm going to introduce you to the conceptual framework that we use to think about this problem, because it'll frame and help help you understand the remainder of the talk in light of these steps here. So in order for a pathogen to make its way along the pathway, all of these steps have to occur, as I said before. First, an angler has to use live bait fish. They have to purchase live bait fish. They have to purchase an infected fish, and they have to then release that infected fish. If each of those steps in the pathway occurs, that's something we're considering a risky trip, or a trip that results in the release of an infected fish into the water. But if at any point along this pathway, the angler either does not use live bait fish, maybe they use fish that aren't susceptible to the pathogen, maybe they don't happen to buy any that are infected, or maybe they don't release. Any one of those steps going wrong means that the trip is not risky. And all of these things have to go right in order for the trip to have all the dots connected and a pathogen to move along the pathway. So the way we're conceptualizing this helps us to understand the next step in actually quantifying the risk. I'm using a method called stochastic risk modeling. Stochastic refers to the fact that we are using probabilities rather than single point estimates to estimate the risk of a pathogen moving along this pathway. So again, here's our risk pathway. The bait fish gets used by an angler. The angler happens to be using a susceptible fish. They happen to be using a susceptible fish who's infected by the pathogen of interest, and they happen to release that infected fish. All of that has to happen in order for a risky trip to occur. And we think of each of these steps as having a certain probability of occurrence. If you remember back to elementary school math class, it's drawing marbles out of a bag, it's rolling a dice, whatever it is. That event has a particular probability of occurrence. But we want to know what's the probability of activity A and B and C and D occurring. And in order to do that, we multiply the probabilities together. Again, if any one of these probabilities doesn't occur, or if any one of these events does not occur, it becomes a 0 and the whole chain is disrupted. Then we use a software to actually do the work for us and conduct hundreds of thousands of simulations, actually conducting this calculation over and over and over to see how often it occurs. So when we do that, we call each of those runs an iteration. So for example, here I have 5 iterations that I just made up, and each of these rows is one iteration. So as you can see, a 1 is put into the row when that step occurs, and a 0 is put into the row when that step does not occur. So you can see, even though sometimes an angler uses live bait fish, or maybe they use susceptible fish, or maybe they even use an infected fish, that doesn't necessarily mean that there's going to be a risky trip. Every single one of these steps, or every single one of these columns, has to have a 1 in it in order for there to be a risky trip. And what our software actually does is count the number of times that that occurs. So I'll show you that in a little bit when we talk about the results. So in general, we have a number of model inputs. We have a number of parameters that we use, estimated by probabilities, to see what the outputs of interest are going to be. We have a number of outputs that we're interested in. First, we want to see the probability of at least one VHS release occurring in a given year. I want to see the probability of any given trip being risky or resulting in the release of an infected fish. And I want to calculate the number of risky trips occurring in a given year. Today, I'm going to talk about just N risky, the number of risky trips, because of time, but I'm happy to answer questions about some of the other outputs as well. So we have all this data we need to get, and how do we get it? Well, we did an angler survey, and we actually sent out a mailed paper survey to 4,000 anglers across the state. I've talked about this survey before at previous showcases, and I'm not going to spend a ton of time on it today, but please ask me questions if you are able. We used the survey, I'll go back a slide here, to get data from most of our inputs. So as you can see, all these angler behaviors, the probability of using live bait, of using susceptible species, the number of susceptible fish that they bought, the probability of releasing, the number of trips per year, all of that is coming from our angler survey. The probability of a susceptible bait fish being infected with VHS, or our P3, is also known as the prevalence. How common is the virus in the fish species? And we have actually a hypothetical or theoretical data source for that. And I'll tell you about that when I get to that slide. And finally, the number of licensed anglers was taken from historical license data. So as you probably remember from before, I had 3 pathogens that I identified: VHS, Asian fish tapeworm, and ovarian parasite. But today I'm only going to talk about one. I think you'll find it's still plenty of information to get through, but we do plan to do the full simulations for the other 2 pathogens as well. But for today, I'm going to talk about our scenarios for VHS and how we quantified the risk for each one. Just a little bit of background on the VHS in the state of Minnesota. Currently listed as susceptible to VHS in Minnesota are emerald shiners and spot-tail shiners, which make up a small percentage. About 22% of anglers use these fish every year in Minnesota, according to our survey. However, fathead minnows are also considered susceptible by several international and national authorities. So we wanted to model the difference between if fish that were currently tested were infected, or if we added the possibility in the model that fathead minnows could also be infected. So that's those two different scenarios there, whether they're just the shiners or the shiners plus the fathead minnows. Another consideration is where the VHS is located. Currently in the state of Minnesota, there are a number of risk-based zones that determine kind of how much VHS risk there is in each area. And this red highlighted zone up in the upper right here is the Lake Superior watershed, which is considered high risk for VHS because of its proximity to Lake Superior. So we wanted to test the difference between an outbreak that was contained in that region to an outbreak that was happening all across the state. These 2 factors in combination give us the 4 model scenarios that we're going to be talking about today. The first model is a statewide outbreak with only currently tested species. The second model would be a statewide outbreak where all susceptible species, including fathead minnows, are allowed to, or they have the possibility of getting the virus in the model. The third model would be an outbreak in that zone 3 or that Lake Superior watershed, only in the currently tested species. And finally, a zone 3 outbreak in all the susceptible species would be model number 4. So I'll compare the results of these models as we go through. But before we do the results, we have to talk about the risky behaviors. I'm going to move fairly quickly through these slides here, but please ask questions afterwards if you're interested. So first, the probability of using live baitfish. According to our angler survey, about 70% of anglers said that they used live baitfish, and of those, only about 6% said that they used from zone 3. So in those models where the outbreak is contained to zone 3, if an angler is not getting bait from that zone, they're not considered risky because the outbreak is contained. But for those that are in the statewide model outbreak scenario, they could be getting bait from any region, and it's equally likely that they would be getting a VHS positive fish from there. Secondly, we looked at the probability of using susceptible fish. Remember that I said that emerald and spot-tail shiners are the 2 species considered susceptible to VHS at this time. And so they're considered in the model there. And as I said, about 22% of anglers use those species. And they buy an approximate number, about 20 fish, but it's a highly skewed distribution with some anglers buying hundreds of shiners at a time. In the second category of models, however, though, we added the possibility of fathead minnows being infected by VHS. They're not currently tested in the state of Minnesota, but they are listed as susceptible by the International Fish Health Authority. As a result, we thought it would be interesting to see what happens if we allow them to be positive for VHS in our model. And they're used by an overwhelming 75% of anglers. So that you can imagine what happens when you allow fathead minnows to get VHS, but we'll see. Next, we wanted to figure out what the probability of infection with VHS is. And this one is the one that I said we're going to get a little bit theoretical. It's a little complicated because currently in the state of Minnesota, we assume that there's no VHS. We have not had a positive inland water, despite hundreds of tests being conducted every year. And for the last 10 years, we haven't had positive inland waters. But we wanted to see what would happen and what would be a likely scenario if the pathogen were to make its way into the bait supply in Minnesota. What would be a reasonable assumption about that? And the way we thought about it was in terms of detection probabilities. Under the current testing regimen for VHS, We're 95% confident that a positive fish will be detected if the prevalence of the disease is above 5%. That's just definitionally how we determine how many fish to take in a sample. And we want to make sure we're at least 95% confident of catching a fairly high presence of the virus. So the way we thought about it was, what's the probability of a particular prevalence going undetected? What's the likelihood that we're missing it? And this is what we found. So this blue line here represents the probability of detection in a standard 60 fish sample. And along the x-axis is the theoretical pathogen prevalence. So as you can see, the likelihood of having a pathogen at really high levels and going undetected is pretty low. We're 99.9999% likely to detect VHS at 61% prevalence. So that's pretty good. But what about if the prevalence is pretty low? What if the prevalence is 11%? Well, in that case, we have a 99.9% chance of detecting it. Still pretty good. But if pathogen prevalence is 1%, we're only 50% likely to detect VHS. It means we are just as likely to miss VHS at 1% prevalence in the bait supply than we are to detect it. So the reason we incorporated this variability into our model is we wanted to account for the fact that our test is imperfect, and it's possible, unlikely, but it is possible that we're missing small amounts of VHS positive fish moving through the bait supply, even under current testing regimens. So these are the values we used for prevalence in our model. Finally, we wanted to look at the probability of releasing live bait fish. Most anglers properly dispose of their bait in the water. Awesome, we love to see that. But unfortunately, about 20% of anglers said that they released their live bait into the water at least some of the time. Some of the other smaller scale surveys that we've done and some work by the DNR suggest that that number may be even higher, up to 30 or 35%. But for the purposes of our model, we took a conservative estimate of 20%, or about 1 in 5 anglers releasing their bait into the water. And we also wanted to calculate the number of trips per angler per year. Most anglers have kind of a small number of trips. They probably wish they were getting out more, but fewer than 10 trips per year. But as you can see by the very strong, very long right tail of this distribution, some people are going over 150 times per year. So that's something important to keep in mind for this pathway in general, is most people are going to be down at the bottom, but these long tails allow for some kind of improbable events to start occurring more and more. And you'll see that in just a bit. So before we talk about, So now that we've talked about the actual quantifying of the behaviors, we can put it all together into our quantitative risk model and figure out, what if I want to figure out the likelihood of the first step and the second step and the third step occurring, all things going wrong, to connect the dots and make a risky trip happen. So as I said before, I'm going to show you the number of risky trips that occur in a given year. This graph is kind of complicated, so I want to take just a moment to orient you to it. Each of these colored diagrams here illustrates a different scenario, listed on the left there. So statewide all susceptible is the statewide outbreak with all fish, including fathead minnows, able to be infected with the pathogen. Statewide currently tested is a statewide infection just in emerald and spot-tail shiners. Zone 3, as you can probably guess, is an outbreak centered only in the Lake Superior watershed, with all susceptible species allowing to be infected. And Zone 3 is, the final one is Zone 3 with just currently tested species. The height of these curves here, it's a little difficult to see because they're so stretched out. The height of these curves here indicates the probability of that value occurring. So along the x-axis here, I have the number of risky trips in a year. As you can see, the bulk of these graphs is clustered around 0. And if you look at these numbers I have on the right side, you can see exactly what I mean. In the statewide all susceptible scenario, 88% of our iterations, or 88% of the times we ran the model, there were no risky trips. 0 trips that resulted in the release of an infected baitfish. However, as you can also see, this line extends quite a ways off to the right, all the way up to, I actually truncated the graph at 200,000 because I wanted to be able to visualize. Otherwise, you're talking in the millions of trips occurring every year as a possibility. Now, that only occurred in 1 or 2 iterations each time, but it did occur. So what the model is telling us is that although these no risky trip scenarios are very common, happening in 88% and even 99.8% of trips in some of these scenarios, there's still a possibility for a number of risky trips to occur. And as you can see, even in the lowest, kind of smallest, most contained outbreak scenario down here with the zone 3 currently tested, there was an average of 4,360 risky trips occurring every year across all of these iterations, even though 99.8% of them were not, didn't have any risky trips at all. And that number goes all the way up to 415,000 trips that could result in the release of an infected baitfish happening every year if we were to have a statewide outbreak of VHS that was present in all of the susceptible species. In terms of where this is happening, we did look at where the anglers are saying that they went fishing. And unfortunately, the rate of release is constant across all areas of the state. It doesn't seem like any one group is better or worse, as much as we might want to point fingers. Approximately 20% of anglers were releasing their bait across the state. However, some areas experience greater fishing pressure. It's probably not surprising that the center of a state there with lake country and cabin country sees a lot of fishing. And so there's a lot of release events that happen there. So in conclusion, there's good news and bad news. The good news is that a majority of anglers don't release their bait. And a vast majority of trips, remember it was like 88% in the highest risk scenario and 99.8% in the lowest risk scenario. The vast majority of trips are not likely to be risky, and a vast majority of iterations had 0 risky trips occurring. So most of the time we're going to be okay. Under current conditions, VHS positive fish are unlikely to be released on the landscape. The bad news is, I have my buddy Aristotle here to help me out in explaining it. He says, for what is improbable does happen often, and therefore it is probable that improbable things will happen. And this apparent paradox might seem a little bit esoteric, but I think it explains kind of where we are at in terms of understanding risk of pathogen introduction. Given gaps in our current management framework, the release of VHSV positive fish is possible. And so I have just a few recommendations for reducing the risk of VHSV release into Minnesota waters. First, given the fact that a zone 3 outbreak could occur and there were some risky trips associated with that watershed, one could prohibit bait sourced from high-risk watersheds. Additionally, we could expand VHSV testing to include the susceptible fathead minnow species, as well as other species that are being increasingly identified as possible hosts for VHS. So this would allow us to stop turning a blind eye to 75% of anglers who could be carrying positive fish around. We could also increase the VHSV testing threshold to reduce the probability of VHSV fish being missed. Remember that graph I showed with the probability of detection kind of curving like this? That graph is entirely a function of the number of tests that are currently conducted. And so we could increase that to reduce the probability of VHSV fish being missed. And finally, we could improve angler compliance with existing regulations prohibiting live bait fish release. As I said before, the majority of anglers are following the rules, but 20% of them are not. And if you saw my undergrad UROP students talk or poster yesterday, she talked a little bit about why they aren't. So in terms of future directions, we plan to complete exactly the same risk analyses I just showed you for the 2 other pathogens we identified as high risk, Polypleistophyra variae and Asian fish tapeworm. And importantly, we want to do sensitivity analyses to identify the most important steps in the pathway. Each of these potential management scenarios I outlined here has a certain cost or burden associated with it. So we want to make sure we're getting the most bang for our buck. Without tipping my hand too much as to the results of the sensitivity analysis, it comes down to anglers. Preliminarily, we're seeing that the rate of release is one of the most important steps in determining overall risk, but preliminary. Stay tuned. Our next project with MACERC is going to dive into the social and psychological determinants of bait release behavior and see if these factors differ across different types of anglers to inform more targeted management. so that we can avoid applying a one-size-fits-all management strategy and instead have optimized risk management to ensure that we're getting the anglers that are the most likely to do risky behaviors. With that, I will take any questions, and please let me know if you want to go back on slides or anything like that, but thanks so much for coming.
All right, thank you very much, Meg. Excellent presentation with a lot of information to get through. Virtual round of applause to you.
Thank you.
Just a reminder, please put your questions in the chat box or the Q&A box, and Meg can get through them that way. Well, Meg, it's a pretty quiet group out there this
I guess
afternoon.
so. I got off the hook. No pun intended. One thing I'm curious if people have thoughts on is looking at these numbers here. Sounds really good if 88% of our iterations had no risky trips, but on average, there were 400,000 risky trips. So you kind of get two different risk perceptions out of it. On one end, you say, it's not very likely to happen. But on the other, you say, oh my gosh, on average, 415,000 trips occur. I'd be curious if people saw this talk and thought, oh, that sounds really risky, or if they thought, nah, we're good. Looks like there's one question in the Q&A.
Question that came in from Rachel Crabb. Do you know why people are releasing bait?
Yeah, so that is a great question. We've done a couple of things looking at that. One was when we conducted the survey, we did an open-ended question that asked people, why do you release your bait? And then for a little more focused approach, my undergrad student worked this summer to do a number of open-ended questions where we asked people, what's the advantage and disadvantage of releasing live bait? What's the, what do you like, dislike? What would be reasons that make it easier or harder? And one really interesting thing we found was that everyone who said that they released live bait and responded to our survey admitted that it could be spreading aquatic invasive species and disease. They listed that as a disadvantage or something that they disliked about the behavior. So that led us to the question of, okay, so then why are they doing it? They're aware of the risk. And for a lot of those anglers, it had to do with a lack of bait disposal sites at the launch. They also felt like it was cruel to throw them in the trash. They didn't want to do that. Another factor that came up was, although they perceived the risk to be there, they thought that the spread of AIS and pathogens via watercraft movement or other activities was greater. So there was some kind of pointing fingers at other pathways, and I think that might be a factor. And ultimately, I think another piece is they don't know it's the law. I think it was like almost 80% of the people who said that they threw it in the trash said that they did so because that's the law. And no one who threw it in the water said anything about regulations, the DNR, disapproval from, legal, like they just didn't see it in terms of legal. That wasn't a salient value for them. So it's not that they don't know, it's just that it's not an important factor in their decision.
Megs, it looks like you have a comment here from Wendy, who is responding to your previous statement about 88% of the trips aren't risky, but when it happens, it could be bad. It sounds pretty risky to her.
Yeah, I think it's so interesting because when I first saw 88%, oh my gosh, that's so great. But it can happen. And I think as we see with the global pandemic, even if most of our interactions are safe, sometimes the very small probability but high risk things are, they loom large in our imagination. Thank you for that answer.
Okay, there's a yeah, so we have a comment that came into the chat box from Megan, who makes a good point. This sort of the ethical dilemma of throwing bait fish in the trash. Meanwhile, they put a hook through its head and feed it to a fish is okay.
Yeah.
It's a
Yeah, it is. That always shocks me that they think that that would be an issue. Yeah, I don't fully understand that. I think that there's kind of an idea that when you release the fish, it can go on to like live a fulfilled life and it can contribute to the ecosystem and be forage for other fish. So I think you sort of imagine that to be a very positive and pleasant outcome as opposed to being thrown in the trash, which maybe helps you turn a blind eye to it before.
Okay, Meg, Wendy is wondering, did you model the effect of people changing their behavior?
No, but that's coming up next. So that's kind of the pulling it all together piece of, I talked about the future directions. We're going to see if these behaviors differ across different types of anglers and if different kind of factors of social or psychological reasons factor into their behavior, and then actually model the effect of them changing their behavior in response to some sort of management. By and large, I can tell you, of course, that release behavior, goes down, risk goes down, but this little more complex, how does behavior change affect risk? That's coming up, so stay tuned. Thank you for that question.
So we've got two questions that have come in, both with similar ideas here. The first one, does Minnesota have a certified virus-free bait program as other states?
No, there is a voluntary program that producers and harvesters can take place in that is designed to reduce the risk of bait being contaminated with pathogens or other invasive species like water milfoil or crayfish or Eurasian rough. But again, that's voluntary. That's called AIS HACCP, HACCP, if you're interested in learning about that. But it's not a certified program.
I would add, Meg, that there are the two species that you referenced, emerald shiners and spot-tail shiners, that are susceptible to VHS are required to be tested. The source is required to be tested once per year.
Right. So there is, I can see there's another question about testing. And so there is some testing. It's on those species for VHS once per year. And there are certain regulations and prohibitions for harvesting on VHS positive waters. But for all the other species, including fathead minnows, there is not required testing, and it's not like a lot by lot test. It's not like every time the harvester goes out, they bring a couple fish to get tested. It's a once per year designation on the either the farm or the wild source.
All right, we can give you another minute here if anybody has a last question. Otherwise, we can roll.
I'll put my email up again too if anybody's interested. Yeah, you can grab my email off the screen here or from the website if you have any questions or further thoughts or groundbreaking insights. Please contact me or Nick.
All right, seeing no other questions in the chat box there, we'll go ahead and wrap it up. Thanks again, Meg, for the excellent presentation. And I encourage everybody to come back tomorrow at noon for the day 3 of the MASERC Showcase.